ER, okay, the calcium is going to be released into the cytosol. Then calcium can be taken up into the mitochondria, and then again, water can flow, or water can follow, and then the mitochondria will release the cytochrome C. That's one way it can happen. So to turn that on, again, remember in chapter 4 we talked about how you can have the unfolded protein response, and how the accumulation of unfolded proteins inside the reticular, how that's going to, first of all, send those proteins out into the cytosol to be broken down by the proteasome. Okay? Another thing that can happen is if you have a cell that is accumulating a ton of unfolded proteins, and the proteasome can't even really keep up, that in and of itself signals to the mitochondria that it needs to commit suicide. So it can turn on the apoptotic cascade. Okay? And then to further complicate it, inside the ER there also happens to be a caspase. Now, what is not known is what's actually cleaving caspase 12, but this is a different pathway. So what happens is caspase 12 is activated when the cell is stressed, and this is located inside the ER. And then what happens is that once it's activated, it's able to move out, sort of like with the unfolded protein response, out into the cytosol, and it is able to directly cleave procaspase 9. So there's no requirement there to actually form an apoxosome. Caspase 12 is found in limited quantities, but it's on its own able to cleave caspase 9, and then caspase 9 can go on and activate caspase 3. So that's sort of mitochondria independent triggering of caspase 3. Okay, now last class I mentioned how a killer T cell or an NK cell is able to eliminate cancerous cells or virally infected cells by releasing perforin, which is just going to poke a hole, and then they also release granzyme. Now you can see here granzyme B is serine 38. And so these are just different ways that a killer T cell is able to turn on apoptosis. Now, it's safer for a cell to undergo apoptosis rather than necrosis, regardless of whether there's a virus present or not. Okay. So this granzyme B, the serine 38, is able to directly activate procaspase 3. It's another way to trigger an effective cascade, and that's what's going to allow apoptosis to occur. Okay, so there's different regulators of encourage you to really try and learn the different BCL2 bounding members, which ones are inhibitors and which ones are going to be activators of apoptosis and which will be inhibitors. Okay, so to regulate apoptosis, we have two different inhibitors of apoptosis. Now these were first discovered actually with viruses. So you can imagine that a virus infects one of your host cells and wants that home. Okay, so wants a place to make more copies of the cell. And so as soon as our cells become infected, they're going to want to commit suicide because they don't want to support the growth of the virus. So what a lot of viruses, or a lot of intelligent viruses have learned to do is to make inhibitors of apoptosis. So for example, that's how herpes viruses are so successful, is they have a number of different ways to inhibit apoptosis. So they're going to express these proteins immediately upon entry into your host cell. Now the first inhibitors of apoptosis that were discovered were actually viral proteins. Okay? So we now know that human ones as well, but some of the best studied inhibitors of apoptosis came from viruses. So we have one here, CRAM-A. And this is sort of the first studied inhibitor of apoptosis, and it came from cowpox. And then over here, 35 is just one potential backboulevard. And then once they actually found these inhibitors, then they were able to look for analogous proteins in the mammalian genome. Okay? And the way they work is by negatively regulating pathway activity. They've been broken down again into the IAP or the BCL2 family. Okay, so here we have our BCL2 family. And again, you need to learn the names of these. So you have like the BCL2 family members. Okay? And these are going to be anti-apoptotic. And then you have the group 2, which includes the BAC and the VAC. And those are going to be pro-apoptotic. Now you'll notice the only difference between these two is the presence of this BH4 domain. Everything else is going to be highlighted now, but note here you see that these two groups have a transmembrane domain. Okay, now in group 3, these are pro-apoptotic as well. So you have BID and BAD and BIN. Okay? Now the BID and the BAD are going to lack a transmembrane region, which means they're just going to be present in the cytosol. And it's actually BAD that we're going to talk the most about. So a combination of these different anti- and pro-apoptotic proteins is going to determine whether or not apoptosis occurs. Now generally, inhibition of apoptosis, which is going to allow for cell survival, is going to take place with BCL2 and BCL-XL. Those are the ones you need to remember. Okay, so this is one way to look at it. On the left, we're looking at a normal cell. And so this is a cell where apoptosis is going to be inhibited. And the way that works, what I didn't label on here that I should have shown you, is that up here you're looking at BAD. Okay? So regardless, what you have is you have anti-apoptotic proteins, the BCL2, okay? And those are going to be interacting with a pro-apoptotic protein like BAC or BAC. So long as those are together, essentially your pro-apoptotic protein is going to be occupied by this anti-apoptotic protein. Now the BH3 only protein up at the top is in an inactive form. Okay, so it's not going to be able to do anything. Now as soon as this BH3 only protein receives some sort of a death signal, that is going to cause it to become activated. So the BH3 only protein becomes activated, and then what it does is it binds to your anti-apoptotic protein BCL2. Okay. This is going to release the pro-apoptotic protein, BAC or BAC, and that is going to cause apoptosis. So essentially, this is where you're going to have oligomerization of the BAC or the BAC, and entry of the oligomers into the formation of the pore in the outer membrane of the mitochondria. Does that make sense? You need to remember that your BH3 only protein is in an inactive form, so your BAD is going to be in an inactive form in a normal cell. Okay, so alternatively, here we're looking at the IAP family. Okay, so again, what happens is that when you have your pores in the membrane and your cytochrome C can come out, this is the same diagram here, so cytochrome C is able to emerge from the mitochondria. What can happen is you also have these other proteins that are released. Okay? So we have SMAC and Diablo, and these are going to be the inhibitors of the IAP. Now, what you need to highlight on here is that your IAP is able to directly interact with a component of the apoptosome. So if the IAP inhibits either the apoptosome formation or it inhibits the apoptosome function. So it's either going to prevent your cytochrome C from binding and turning on the apop, or it can inhibit your caspase 9 formation, or it can inhibit the whole apoptosome. So when cytochrome C is released, you'll note that SMAP and Diablo can also be released. So cytochrome C is able to form the apoptosome, and SMAP and Diablo are able to inhibit the inhibitor of apogensis. So it's like a two-pronged effect. So you have to note that it's not just cytochrome C that has to come out of the mitochondria. You also have to have release of molecules that will inhibit the inhibition of apogensis. Okay, so the overview here. Again, you have your apoptotic death signal. You're going to have activation of BAD, the BH3-only protein. This is going to cause activation of your BCL2 protein. You're then able to form a pore on the mitochondrial membrane. Again, cytochrome C is going to be released that will form the apoptosome, ultimately turning your inactive caspase 9 into active caspase. Okay? Note, you're also going to release SMAD, of course, this should be SMAP and Diablo. Okay? So you have inhibitors of the IAP protein. Okay, so once you have your initiator caspase, you're going to first turn on your caspase 3. You're going to have degradation of protein. Okay, all these different targets of the caspases. You're going to have DNA fragmentation. And then we're going to form those apoptotic bodies. So in order to effectively get rid of those apoptotic bodies, there has to be some sort of a phagocytic cell that can come along and can eat those leads. So this is just showing that you have phosphatidylserine in a healthy cell found on the cytosolic reflex, and then after apoptosis or during apoptosis, those phosphatidylserine are going to be displayed on the extracellular tissue. Now, on macrophages, there are receptors for phosphatidylserine, and so that is just going to be receptor-mediated phagocytosis. Okay, so what are the different pathways that are going to turn on cell survival? This is where we get back into the signaling. Okay, so over here we're just looking at different kinds of receptor tyrosine kinases that can turn on cell survival. So again, all cells are inherently wanting to die or have a pathway to death, but then we can, using different signaling pathways, can allow for cell survival and in fact cell division. Okay, so through receptor tyrosine kinases, we talked about the MAP kinase pathway. So there's another pathway here going through PI3 kinase. What it can actually trigger is it can trigger AKT. You guys talked about this in biochem? Talked about protein kinase B. Okay, so AKT is just another word for PKB. Okay? So you have your PKB, so you have a kinase that is able to phosphorylate a number of different substrates. Okay? So the first thing that it can phosphorylate is BAD. So AKT will phosphorylate and therefore inactivate BAD. There's other kinases that it can phosphorylate. Regardless, once you have phosphorylation of these proteins, they're actually going to remain in the cytosol, sequestered away. Okay, now over here on the left, so if AKT is going to phosphorylate and inactivate your pro-apoptotic protein, it's also able to phosphorylate and activate anti-apoptotic protein. Okay? So PREB is a transcription factor, and so it's going to phosphorylate and activate PREB. And it can also turn on the transcription factor NF-κB. Okay, so on the right, we have our receptor tyrosine kinase. And you can see through an SH2 domain containing protein, there's recruitment of DRAP2, and then SOS, again, the same pathway that I talked about before. Okay, so you're going through the MAP kinase pathway. RAS is phosphorylated and activating RAS and MET, and then MAP kinase, or ERK. Okay? So what happens here is that ERK is able to also phosphorylate and therefore inactivate BAD. And so in this case, BAD becomes phosphorylated and is sequestered in the cytosol. It is also, ERK, the MAP kinase, is also able to phosphorylate for caspase 9. So this is before it's actually going to turn on caspase 3. So basically, by phosphorylating for caspase 9, it is going to inhibit for caspase 9's activation. And then finally, we have activation of CREB. And again, CREB is going to be your transcription factor, and you're going to have transcription anti-apoptotic protein.
Question.
Yes.
Now, can you tell me again the purpose of that?
So this is where first of all, you should know that BAD is it pro-apoptotic or anti-apoptotic? It's pro-apoptotic, okay, and it is present in the cytosol. It's the BH3 only domain only containing BCL2 membrane. Okay, so there's your BAD. And normally what BAD does, here's your BAD in here. So when BAD becomes activated, it is going to turn on apoptosis, pro-apoptotic, and it does so by binding to BCL2. I think the easiest way to look at this is actually, again, not in the textbook. Okay, there's one thing that's missing on here that I think that would make it easier for you guys to follow is on the right, are we inhibiting apoptosis or are we allowing apoptosis to happen?
Allowing.
On the right, we have inhibition of apoptosis.
Inhibition.
Yeah. So you can, however you want to put it most simply for yourself, self-survival, cell-live, whatever you want to say. That's what you're saying on the right. Okay? And this goes back to where we started Monday. We were talking about trophic factors. Okay? So, generally, cells, if they receive a trophic factor, that's going to be some sort of a signaling molecule from another cell that is going to allow the cell to survive. So make sure, just for when you're going through this, make sure that you recognize that on the right in this case, you're looking at cell survival. Okay, so this is how it works. We'll start with that. What happens is the cell at the top here receives the trophic factor. It's going to be a signaling cascade. The chances are, what kind of receptor is this? Receptor tyrosine kinase. Okay? So it's going to receive this cell survival signal, and you're going to turn on one of the different pathways. In this case, we're turning on the PI3 kinase pathway. Okay, so regardless, you're going to you're going to turn on your kinase PKB. So that's just AKT again. Different textbooks use the different names. Okay, so here we have our protein kinase B. So what happens, as we said, is that your PKB is going to phosphorylate your BAD. Okay, so next here you see what BAD is actually doing. So the red ball there, the BAD becomes phosphorylated. And as a result of the phosphorylation of the BAD, it is going to stay in the cytosol. Exact same thing that we saw here. BAD becomes phosphorylated, it's sequestered. Okay? So if you have BAD, and it remains in the cytosol, it comes off of the BCL2 family. Okay? Now, the other BCL family members have that transmembrane region. And so we are looking here at the membrane, the outer mitochondrial membrane. Now, present in the membrane, you have BAD. Now, is that pro-apoptotic or anti-apoptotic? Right, so that's pro-apoptotic, and it is found in the membrane. And then you have the BCL2 family member, also in the membrane. So that's just where they're always found. So if we go back here and take a look, in a normal healthy cell, you have your anti-apoptotic protein and your pro-apoptotic protein found together. Okay, so there's your BCL family member, and in the absence of that, what happens is your anti-apoptotic proteins are going to inhibit your the back oligomerization. Okay, therefore, cytochrome C is going to stay inside the cell, inside the mitochondria. Okay, so let's look at what happens and how we can actually turn on pathway activation. So in this case, we are looking at turning on a cell death. There's no signaling from there's no trophic factor to turn on cell survival. Therefore, the PI3 kinase pathway is not turned on, PKB is not turned on. Okay, so what do we have? We have our BAD, and we have our BCL anti-apoptotic protein bound together. Okay. As a result, your BAD is going to be active, it's going to allow iron to move into the mitochondria and water as well. Note, cytochrome C is going to flow up. Cytochrome C goes up, forms the apoxosome with APAS1 and procaspase 9. This is going to allow the cleavage of procaspase 9 into active caspase 9. So now your initiator caspase is going to turn on the breakdown of procaspase 3 into active caspase 3. The only thing that's wrong with this is there should be an arrow here showing that caspase 3 in its active form is able to cleave the substrate, resulting in cell death. Okay? Does that make sense? Okay, so why is apoptosis significant? Okay, so basically two opposite extremes to look at. First of all, if apoptosis doesn't work. Okay, so this is where if you don't have cell death, you just think you're going to have overgrowth of cells, you think of a transformed phenotype, and ultimately different kinds of cancer. Okay. So, if you have increased BCL2 expression, so increased expression of your anti-apoptotic protein, this is seen in a number of cancers. And looking at the amount of the BCL2 is going to actually be sort of a predictive factor for different kinds of cancers. So that's one of the things that is often measured, is the quantity of BCL2 that is found in these different in samples from these different cancers. Okay. There's different kinds of chemotherapeutic drugs that specifically target and activate the apoptosis machinery. It's a difficult balance to make sure that you're only targeting the cells that you actually want to purify and not other cells as well. And then finally we see this essentially a type of interfering RNA. So one of the thoughts is to try and interfere with the inhibitors of apoptosis. And that would just be a way to try and deal with the cancer. Okay, and the opposite extreme is when you have too much apoptosis. So premature starts of apoptosis. And this is when you're going to end up with undesirable cell death. So we mentioned at the beginning about how apoptosis is responsible during the T-cell development and for learning what belongs to self and what is not self. Okay, so it's like biocyte education. So normally what happens is in your immune system you're going to have T-cells that see healthy cells and they recognize them too strongly. You don't want to keep around T-cells that recognize their own cells strongly. So those are going to be eliminated. Alternatively, T cells are also eliminated if they don't recognize your cell at all. So there's a balance between recognizing what belongs to you and not over-recognizing it. And that's just what's called lymphocyte education. And so, if for whatever reason that education during embryonic development doesn't occur properly, then what happens is individuals will end up with different sort of autoimmune conditions. Now, when you have all these different lymphocytes and you have all these different T cells clones, if they start to expand, they can expand at any point in their life. And when they start to expand and they overgrow, you have clonal expansion of this one particular type. This is when you end up with your autoimmune diseases. Okay? But what's interesting about those autoimmune diseases is that is all determined based on whether or not the appropriate T cell clone dies during thymocyte development. So there's just a list here of different types of autoimmune diseases that have actually formally been shown to involve failure of these different apoptotic pathways during development. Yes. No. Okay. So obviously, this lecture on apoptosis is what, 38 slides long? Yeah. But just keep it complete. You have to make sure that you read the little paragraphs about the different kinds of cell death. Okay?
Yeah. What?
Which paragraph?
Question?
Which paragraph? From the text.
Which paragraph?
You guys read your textbook? Sorry. Someone just said what paragraph? In the textbook. There's a textbook for this.
Yeah, better. Yes, better.
Okay.
So now we have to go to the
Okay, so really simply you have autophagic cell death. You guys should always remember that autophagy is just cell eating.
I'm going to show you
Okay.
the tablet.
So the type 2 program cell death is, again, just go back to the pictures at the beginning of the lecture that I put up for you. Last lecture, where you can actually see those large vesicles inside the cytosol. It's going to be characteristic of autophagy. Okay? And this is really the only time that you actually see the actual consumer cells like this. So you have increased lysosomal activity, you kind of get all the different enzymes in the presence of the components inside the lysosome. And then finally, you have non-lysosomal cell death. Okay, so this is where you don't actually have the characteristics of apoptosis, but what you have is you have swelling of your different organelles. Okay, but in this case, you're not actually going to see lysosomal digestion. And then just so we can keep it complete, anointis is just a type of cell death which is dependent on or turned on by different extracellular components, so anchorage-dependent cells from the surrounding extracellular matrix. Okay, so again, cells like to touch each other. They like to form those monolayers into a single layer. And when they lose that contact inhibition, it's often should indicate that a cell wants to undergo a type of cell death. So when cells are going to detach from that extracellular matrix and an underlying extracellular matrix, they often undergo anointis. And then excitotoxicity, and this is specific to the nerve cells. And this is basically saying when they are damaged, and then ultimately going to be killed. And that just comes from hyperstimulation of those nerve cells.
Yeah.
Well, amazing.
And you have to watch
Question?
the question.
No.
Oh, you want a question?
Sample question. Yes.
You make them transcribe it. Very quiet.
Something I put down for the commercial, really?
No? Oh, no.
See? 2 hours, not bad.